Welcome class. So our new lecture is on autosomal recessive condition and we have started genetics chapter. So today we will be talking about the autosomal recessive conditions and their genetics as well. So there is a mnemonic I have so I am going to share it with you. It's called as autosomal disease involve a recessive white cyst on my big hand. Suppose here is a hand, okay, and there is a white cyst. From here till here, I have described it in the previous lecture. These are autosomal dominant condition. Okay, you can check it there. Today we will be talking about the recessive white cyst. This cyst, okay. A recessive white cyst is actually a recessive. All the autosomal recessive disorders, they can be memorized with a simple white cyst. Okay, A for alpha 1 antitrypsin deficiency, W for Wilson disease, and C for congenital adrenal hyperplasia, and CY for cystic fibrosis, and S for sickle cell, and the last T is for beta thalassemia. Okay, beta thalassemia. This is a simple mnemonic, and now we are going to talk about the genetics. Okay, so let me give you an example of a father and a mother. So one thing is there, one condition is actually there. They both have to be carrier. Okay, like AA. This capital A denotes the defective gene, and this the small letter denotes the normal gene. Okay, and suppose a mother is also having a defective gene and an normal gene. So when these two combine together, they will form AA, which is homozygous A. Now, whenever two capital letters combine together, are transferred to their child, they will be defective. Okay, they will be defective or abnormal. When A and A combine together, uh, they combine together to form A capital and small capital A. So this is a carrier. Okay. Now, when small a and small a combine together, they will form the normal child. Okay, normal child. And when this capital A and the last A combine together, they will form AA, which is again a carrier state. Okay. Now you can see here that only 25% are normal and 50% are carrier, AA are carrier, 50% are carrier, and 25% are defective. Okay. Now remember, when capital A are transferred to their child, so the child will be defective. But if one single defective gene is transferred to their children, those children will be called as carrier. Okay, and the small letter denotes the normal patients. So this will be the percentage in a autosomal recessive diseases. Okay, now the problem, there are 2 points to be noted. One is the two homozygous defective genes or alleles are required to cause the disease, as I showed here. Whenever two capital A's are transferred to the children, then this will cause disease, like here I have described. And if one defective gene is present in the children, that will be called as carrier. Okay, whenever one gene is there, so it makes a carrier. So male and female sex doesn't matter. Any parent can transfer disease to any of the children regardless of the sex because these are autosomal condition. So as I have previously described in my previous video, so whatever the sex may be, now here the father, again I will tell you father or mother. Okay, so sex doesn't matter. That's why we are not using XY as an example. We are just using AA and A because these are autosomal. And autosome means body forming chromosome. Okay, body forming like the eyes, the kidneys, bones, skeleton, everything. Okay, so these are these doesn't differ, okay, between the sexes. Every male and female, both sexes have all these body forming chromosome, like, okay, the 22 pairs. So that's why we are not denoting by the X and Y. So again, if I tell you AA, A, AA, A, A, and lastly, it will form the, so this one is normal, and these two will form the carrier 50%, and 50% is defective. So 25, 50, and 25 will be the percentage. Okay, that's it for now.